Okay. Thermotoga. Thermotoga is, again, a very small group. This is it. And most of these are only one or a few species. There are a few species of Thermotoga, a few species of Thermocypho and Thermotoga. The rest of these are single species. This is a really small group of organisms, but they seem to be quite abundant in hydrothermal environments, particularly deep-sea hydrothermal vents. And Thermotoga maritima is one of the best studied extreme thermophiles there is. These things are all thermophilic. Bifidobacterium less so. It's in the 65-degree temperature range. That's still plenty hot. Thermotoga grows at 90 degrees. These things are heterotrophs. So some of them they all have a bunch of pumps on their outer membrane. Many of them are saccharolytic. They like sugars. Others of them like peptides and eat amino acids for a living. So they're growing heterotrophically by fermentation. And so they're growing by, they are not using electron transport, they're growing by substrate level phosphorylation. They can often use protons or elemental sulfur as an electron acceptor, iron in some cases, generally making CO2 and organic compounds. These things will generate hydrogen for a living by dumping electrons on protons. That's not a very good way to make a living in many cases. A lot of times those organisms are really, really sensitive. That reaction is only favorable if you've got another organism present to get rid of that hydrogen somehow. So you've got to keep the concentrations of hydrogen excruciatingly low in order to grow this way. The morphological trait that's distinctive for this group is a big paraplasmic space. So this is Thermotoga maritima. Here's the inner membrane, and the cytoplasm is inside. Here's the outer membrane. And so this is all paraplasm here. And it's this really loose outer membrane that gives these things their name, the toga name. It really does mean like a loose outer garment, right? These things are riddled with porins and organic compound pumps and that sort of thing to pull in nutrients from the environment. But the function of this large paraplasm is really pretty unclear. You don't need a paraplasm like that for hydrolyzing peptides or sugars or anything. Lots of organisms do that directly by importing and hydrolysis in the cytoplasm. So the function of that is really unclear. These things, because they're heterotrophs like that, they usually grow in organic-rich environments, things like soils, hydrothermally heated soils and sediments. Thermotoga maritima has been studied to death. It's easy to grow. The genome was one of the first genome sequences available. This particular species has terminal flagella, although many related species don't. Thermotoga maritima lacks the electron transport chain, so it grows exclusively by fermentation. Now this creates a quandary for the organism because it's got lots and lots of active transport, right? It's sucking in sugar and amino acids from the environment. How does it do that if it doesn't have an electron transport chain? Because in order to pump that stuff in, you need a proton gradient. How do you make a proton gradient without active transport or without the electron transport chain? The answer is ATPase. Now, if you don't have a proton gradient, you don't need ATPase, right? ATPase is driven by the proton gradient. If you don't make a proton gradient, you can't make ATP that way. You make it by substrate level phosphorylation. But notice the name ATPase. ATPase was named historically for what we normally think of as the backwards reaction, right? Hydrolysis of ATP. Not the synthesis of ATP, but the hydrolysis. In organisms that lack electron transport chains, like Thermotoga, ATPase runs in the direction that the name implies. It hydrolyzes ATP. And if you think about the reverse reaction of a typical ATPase, if you're hydrolyzing ATP, that means you're pumping protons out. And That's how they make the proton gradient, not by electron transport, but by directly pumping it out at the expense of ATP. And then that proton gradient is harvested in order to run active transport to suck in all the organic compounds the organism needs to live. Thermotogae maritima has been focused on by most people who do thermophilic research because it is so easy to grow. Crystallographers love it. If you want to solve the structure of a protein, you have to crystallize it, right? This is how proteins are solved by X-ray diffraction. It turns out that enzymes of proteins from thermophilic bacteria crystallize really easily. At the moderate temperatures that you grow crystals, these proteins are all locked into the lowest energy minimum state. They're not very flexible, and so they crystallize into nice orderly arrays that diffract X-rays really, really well. As opposed to enzymes from mesophiles, which are squishier at room temperature and don't form orderly crystals as easily. And so if you do a PubMed search on Thermotogae, what you find are a bunch of crystal structures and proteins from the organism. We're going to talk later about evidence in the genome of Thermotogae maritima for lateral transfer. It's absolutely clear that 5% of the genes in this organism have come from somewhere else recently. It's a repository of genes from lots of different kinds of organisms. The original paper argues that 24%, I think it is, of the genome was acquired horizontally. This clearly is not the case. But it does provide lots of evidence for horizontal gene transfer. This is a cool one, Thermocyphoafarensis. This is a filamentous organism, and here are the togas, and so their cells connected end to end. That's a septum, that's a septum, that's a septum, and the togas are here. This thing grows optimally at 75 degrees. It actually grows pretty well at 37 as well. Isolated from heated soil in Africa. And for a bit of bacterium Icelandicum, this one's really interesting. So the organism is rod-shaped, and the toga is all at one end, so it forms this giant nodule at the end of the cell. And these things grow at a relatively low temperature, in the 50s and 60s.